Hello everyone, welcome to my channel Deep Learning Biology. Within this video, we are going to learn about cell fusion. You must be knowing what is a cell. Cell is the fundamental unit of life. Each and every organism, whether it's plants, whether are animals, both are made up of cell. Now here you must be knowing every cell it is going to maintain its integrity. Every cell is going to have plasma membrane. Some of the cell they are going to have cell wall, and each cell they are not going to fuse. But within this video we are going to learn one fundamental concept, that is cell fusion. Now here you must be knowing there are two types of cells which are being found within your body. One is germ cell and another is somatic cell. And you must be knowing what are somatic cells. Somatic cells are the cells which are not germ cells. The cells within your body, if you exclude the germ cells, the cells excluding germ cells are known as the somatic cells. And hopefully you know very well the germ cells, they are responsible for making gametes. Now here if you see your body plan, it is just made up of somatic cells. And here within this video I just want to tell you that somatic cell fusion, it can be of two types. One is homotypic cell fusion and another is heterotypic cell fusion. So here you know homo means same type, hetero means different types. If you are going to have cell fusion between same type of cell, for example, if you are going to have spleen cells and if those spleen cells they are going to fuse, that type of cell fusion it is known as homotypic cell fusion. If you see the heterotypic cell fusion, in this case what is going to happen, two different types of cell. Let's take the example spleen cell and the liver cell, they are going to fuse. Such type of cell fusion it is known as heterotypic cell fusion. Now here I just want to tell you, in the case of homotypic cell fusion, what is going to happen, two same type of cells they are going to fuse. So here if you see there can be two conditions. One, after the cell fusion the nucleus may also fuse. So when you are going to see both fusion of the cell as well as the fusion of the nucleus, then in this situation you are going to see the formation of synkaryon. Hopefully you know what is karyon. Karyon is nucleus. So in a synkaryon, the two cells they have fused, their nucleus they have fused. While in the case of syncytium, what is going to happen? The two cells they have fused, but their nucleus have not fused. So hopefully it is clear to you what is syncytium and what is synkaryon. Same way if you see in the case of heterotypic cell fusion, what is going to happen in the case of heterotypic cell fusion, two different types of cells they are going to fuse, and here you are also going to see the formation of syncytium and synkaryon. In the case of syncytium, what is going to happen, the two different kinds of cells they are going to fuse, but there is no fusion of nucleus. But in case of synkaryon, what you are going to see, you are going to see that the two different types of cells they are going to fuse, as well as the two different nucleus they are also going to fuse. So this is synkaryon and syncytium. Hopefully it is clear to you what is homotypic cell fusion and what is heterotypic cell fusion. Now here the most important point I just want to tell you, cell fusion it is going to be a rare event. It is not going to occur frequently. Every cell it has to maintain its integrity. But the cell fusion process you can see during the time of developmental stages. So when you are going to see that the zygote it is going to develop and it is going through different stages, what is going to happen, the cells they are going to fuse. Same way during the time of tissue repair, when you are going to have some wound, at that time also what is going to happen, that wound has to be healed, and that time also what can happen, the cells they can fuse. So always remember, during different developmental stages as well as during the time of tissue repair, what can happen, cell fusion can occur. But the most important point here, you must remember, the process of cell fusion, it is going to be a rare event. It is not going to occur frequently. I just want to tell you, the somatic cell fusion process, as I have told you, it is going to be a rare event. But this event can be induced within a test tube if we are going to add one chemical. The chemical is polyethylene glycol. Here PEG, it stands for polyethylene glycol. Or what we are going to use, UV inactivated Sendai virus. If you are going to mix two different type of cell, and if you add polyethylene glycol or if you add UV inactivated Sendai virus, what is going to happen? The somatic cell fusion process, which is actually a rare event, that will happen at a faster rate. Now here, what we are going to learn about a technology which is known as hybridoma technology. And in the case of hybridoma technology, what we actually do, we take myeloma cell, and hopefully you know very well, the myeloma cells, they are cancerous in nature. Means such type of cells, they are going to have indefinite growth. You know what are cancerous cells? The cancerous cells, they will keep dividing. Now if you are going to take spleen cells, they are normal cells. And normal cells, yes, they are also going to divide, but they are not going to divide as frequently like cancerous cells. So normal cells, they are going to have some definite growth. So in the case of hybridoma technology, what we are going to do, we are going to fuse spleen cell with the myeloma cell. Now within this technology, we have to screen the fused cell. Now here within this video, we are going to learn how we are going to screen the fused cell. Now let's take the example here. Yes, we have taken spleen cell and the myeloma cell. We fuse these cells in the presence of polyethylene glycol. One condition, what can happen, both type of cells, they are not going to fuse. Spleen cells, they are not going to fuse. Myeloma cells, they are not going to fuse. But this chemical polyethylene glycol may induce the fusion of cell. So what are the possibility? Two spleen cells, they are going to fuse. Two myeloma cells, they are going to fuse. Or it may happen spleen cell and the myeloma cells, they are going to fuse. So when spleen cell and the myeloma cells, they are going to fuse, you are going to see the formation of hybrid cell. Now we have to screen these hybrid cell. Now here another important point I just want to tell you. If you look into a cell, you are going to have nucleotide synthesis pathway. The nucleotide synthesis pathway within the cell, it can be two types. One, it can be de novo pathway. Another, it can be salvage pathway. Here I just want to tell you, in the case of de novo pathway, the cell, it is going to synthesize nucleotides, purine nucleotides and pyrimidine nucleotides from the beginning. Okay, so it is going to start from the skeleton of amino acid, carbon dioxide, okay, and there is a slightly lengthy process, and by using that process, cell is going to synthesize nucleotides. But in the case of salvage pathway, what cell is going to use? Cell is going to use intermediates of nucleotide metabolism. So try to think here, by salvage pathway, the intermediates of the nucleotide metabolism, they will be used to synthesize the nucleotides, while by the way of de novo pathway, cell, it is going to synthesize nucleotides, and that is going to happen from the beginning, from the small precursors. So here you must remember there are two pathways. One is de novo pathway, another is the salvage pathway. Now here the spleen cells, they are normal cells. As I have told you, they are not going to divide indefinitely. And if you see the genotype of spleen cell, they are going to have the genotype HGPRT plus and TK plus. Now try to see here. The nucleotide synthesis pathway, it can occur by two pathways. One is salvage pathway and another is de novo pathway. For salvage pathway, there is a need of enzymes like HGPRT and TK Okay, so when you are going to have this enzyme hypoxanthine guanine phosphoribosyl transferase and thymidine kinase, so in that situation what is going to happen? Cell, it can synthesize nucleotide by the salvage pathway. If you see the case of myeloma cell, so here the myeloma cells what we have used, they are cancerous in nature. They can divide indefinitely, but their genotype is HGPRT minus and TK minus. Means they are going to lack enzymes hypoxanthine guanine phosphoribosyl transferase and thymidine kinase. Means in the cancerous cell the salvage pathway is not operational. So here in the cancerous cell the nucleotide synthesis process, it cannot occur by salvage pathway, but they can synthesize nucleotide by the de novo pathway. So here you must remember, in the case of normal cells, spleen cells, both the pathway they are operational, but in the case of cancerous cell, as they are going to lack HGPRT and TK enzyme, the salvage pathway is not operational. Now here what we are going to use, we are going to use a media, and that media is known as hot media. Within the hot media, you are going to have chemicals, the nutrients like hypoxanthine, aminopterin and thymidine. Now here you must remember, aminopterin is a drug which is going to block de novo pathway. So here you must remember, aminopterin, it is going to block the synthesis of nucleotides, and it is going to block the synthesis of nucleotides. It blocks de novo pathway. We are going to discuss what is the role of hypoxanthine and thymidine. Now here I just want to tell you, hypoxanthine and thymidine, they are actually the intermediates of nucleotide metabolism. And I have told you, the intermediates of nucleotide metabolism, they can be used by the salvage pathway for synthesis of nucleotides. The condition is that the cell must have the enzymes HGPRT and thymidine kinase. Now here I just want to tell you, in the cell where you are going to have fusion of two spleen cells, so here if two normal spleen cells they are going to fuse, yes, both of the cells they are going to have the genotype HGPRT plus TK plus, means the fused cells they are going to have the enzymes hypoxanthine guanine phosphoribosyl transferase and thymidine kinase. Now if you are going to put these cells within the hot media, so aminopterin, it is going to block the de novo pathway. But here, as they are going to have this enzyme HGPRT and TK, the salvage pathway will be operational. So in this condition, what is going to happen? Cell, they can synthesize, the fused cells, they can synthesize nucleotides by the salvage pathway. But here I just want to remind you, the spleen cell, normal cells, they are not going to have indefinite growth. They will not grow indefinitely. So these fused cells, as they are not cancerous in nature, they are not going to grow indefinitely. So these cells, they will be ruled out when you are going to perform the subsequent subculture. Next, if you are going to see the myeloma cell, so if you are going to have fusion of myeloma cell, yes, I have told you that myeloma cell, they are going to lack the enzyme HGP, RT and TK. So in this situation, what is going to happen if you are going to put these cells within the hot media? De novo pathway, it is being blocked by the aminopterin. Aminopterin will block the de novo pathway. Same way as these cells, they are going to lack both of these enzymes, the salvage pathway is also not operational. In this situation, what is going to happen? The cell, yes, these are cancerous cells. They have to multiply, they have to divide, and they have to make the copy of their DNA. So they need a very high amount of nucleotides. But here they are unable to synthesize nucleotides by both of the pathway. So both of the pathway, they are not operational. So in this situation, these cells, they are not going to survive. Now let us take the next condition. In this situation, when you are going to have fusion of spleen cell and the myeloma cell. So here I have told you the spleen cells, they are HGPRT plus and TK plus. So the few cells, they are, yes, cancerous in nature, but they are also going to have the enzyme hypoxanthine guanine phosphoribosyl transferase and thymidine kinase. Now when you are going to put these cells within the hot media, yes, within these cells, de novo pathway is not operational, but salvage pathway is operational. So what is going to happen? These cells, they are going to survive. So try to think here. After the event of cell fusion, only the hybrid cells, they are going to survive. Rest of the cells, they are not going to survive within the hot media. Now try to think here. After the event of cell fusion, only the hybrid cells, they are going to survive. Rest of the cells, they are not going to survive. So by using hybridoma technology and by using this hard selection media, you can screen only the hybrid cell. Only the hybrid cell, they will be able to grow, and those cells you can screen. So hopefully you have learned clearly what is cell fusion, what are different types of cell fusion process, and what is hybridoma technology. And within this video we have learned about the hot selection method. Thank you for watching this. Please do like, share the video and subscribe to my channel Deep Learning Biology.